Good evening, and welcome to Channel 4 News. Today, on October 15th, 2014, we remember a great man, Arthur Kornberg. Kornberg himself discovered DNA polymerase. He was born on March 3rd, 1918. He was an American biochemist who won the Nobel Prize in Physiology and Molecular Structures in 1959 for his discovery of the mechanisms in the biological synthesis of DNA, together with Dr. Severo Ochoa of New York University. He was also awarded the Paul Lewis Award of Enzymes and Chemistry from the American Chemical Society in 1951, along with an LHD degree from Yeshiva University in 1951, and the National Medal of Science in 1979. Smart man. DNA polymerase is an enzyme that helps create DNA in the molecules by assembling nucleotides, the building blocks of DNA. The enzymes are essential to DNA replication and usually work in pairs to create two identical strands from a single original DNA molecule. During this process, the DNA polymerase reads the existing DNA strands to create two new strands that match each of the existing ones. Every time a cell divides, polymerase is required to help duplicate the cell's DNA as a copy of the original DNA molecule, which can be passed to each of the daughter cells. In this way, genetic information is transmitted from generation to generation. Before replication occurs, an enzyme called helicase straightens the DNA molecule from its double helix form. Then, the DNA is pulled apart. This gives two single strands of DNA that can be used as templates for replication. DNA polymerase moves along a single strand of DNA, building the complementary strand as it goes. The two-stranded molecule passes through the DNA polymerase molecule after synthesis is complete. If the wrong base is inserted, then the bond is unstable. Because the double strand is passing through the DNA polymerase, the missing or erroneous base can be detected and replaced with ease. The replacement is done by a different part of the enzyme. If DNA polymerase did use single-stranded DNA as a template, and the completed double strand did not continue to interact with the enzyme after synthesis, then the number of errors in DNA replication would be much higher. Now, here's a quick user-contributed video on YouTube from the National Association of Health, Medicine, and Disease.
When the finger domain moves inward, the nucleoside triphosphate is tested for its ability to form a proper base pair with the template nucleotide. When a base pair forms, the active site residues catalyze the covalent addition of the new nucleotide onto the 3' hydroxyl group on the growing strand, and the entire process repeats at speeds up to 500 nucleotides per second. On rare occasions, approximately once every 10,000 nucleotide additions, the polymerase makes an error and incorporates a nucleotide that does not form a proper base pair onto the end of the growing strand. When this occurs, the polymerase changes conformation and transfers the end of the growing strand to a second active site on the polymerase, where the erroneous added nucleotide is removed. The polymerase then flips back to its original conformation, allowing polymerization to continue. As a result, such a self-correcting DNA polymerase will make a mistake only about once every 10 to the 7th to 10 to the 8th nucleotide pairs.
Back to my main point earlier. The main function of DNA polymerase is to make DNA from nucleotides. The DNA copies are created by the pairing of nucleotides to bases present on each strand of the original DNA molecule. This specific pairing always occurs in a very strict order, G to C and T to A. And that is everything you need to know about DNA polymerase and the man who discovered it, Arthur Kornberg. I hope you all enjoyed the program this evening. My name is Jay, and this is Channel 4 News. Good night.